Okay, so the second, class of receptor, that's often found postsynaptically, but is also found, as we stated, presynaptically, where it can influence the presynaptic sending neuron. And also, it can be found on astrocytic membranes around, individual synapses, is something called a metabotropic receptor. And a metabotropic receptor, remember the metabo portion of that name refers to the fact that activation of these metabotropic receptors can really significantly influence the metabolism, the metabolic activity of the cell. And so these metabotropic receptors are really quite interesting. They're very, they have a plethora, like a whole series of potential responses that they can generate. So unlike the ionotropic receptors, remember they are the ones that span the membrane and have a gated channel, right, embedded, as a component of their structure. A neurotransmitter binds to the ionotropic receptor, and the receptor moves, shifts its shape, and opens a hole that allows for sodium or potassium or chloride or something to pass through. That allows for rapid hyperpolarization or rapid depolarization. The metabotropic receptor is structured differently. It has, a portion that spans the membrane from, let's say the outside to the inside. My head's inside. And on the inside portion of the membrane, there is a protein. It's called a G protein. There's many different kinds of G proteins. There's a GI, a GO, a GS, etc. All expressed by different genes, but it's coupled to or attached to this metabotropic receptor on the inside before a neurotransmitter binds. Now, when a neurotransmitter binds, when a ligand comes along and binds to the metabotropic receptor, just like the ionotropic receptor, that complex moves, right? It shifts its shape. But the consequence of shifting shape in a metabotropic receptor is not the opening of a channel, but rather the release of a G protein. So the G protein gets uncoupled from the metabotropic receptor upon the binding of a neurotransmitter. And what's powerful about that is that this G protein, once it's been released by the binding of the neurotransmitter to the metabotropic receptor, can go off and do many things. And in fact, it can do, lots of things inside the cell to impact, the functioning and the metabolism of the cell. So one thing it can do is so a neurotransmitter binds, the coupled G protein is released. It can move over here, and here's another machine over here. It's called a G protein gated channel. So this released G protein can come over, bind on the inside of the membrane here, and open a channel that will allow for, let's say, sodium to go through and, sort of depolarize the cell to some extent, right? Or it could allow for chloride to go through or, potassium to go through. It could have an influence over the charge difference across the membrane, but it's a delay that's involved. And also the fact that you're releasing these G proteins, depending on the amount of, stimulation, it could be a persistent, kind of shift in the polarization. It can be like a longer term sort of depolarization or a longer term hyperpolarization. It can, increase the likelihood or probability that stimulation will generate an action potential or, reduce, for a period of time the opportunity for an action potential. So it can really influence the electrical activity, the excitability of membranes sort of over, longer periods of time. It can modulate that activity. So we're going to see there's a lot of neurotransmitters that act at metabotropic receptors that are known as modulatory neurotransmitters. The other thing that this G protein, when released, can do, it can go over here, open a channel, right? But it can also go off and interact with other chemicals. It can bind to something else and change its shape. And then that thing, which is now known as the second messenger, the second intracellular messenger, the G protein that's been released, by the binding of a neurotransmitter to the metabotropic receptor out here in the cleft. The whole thing moves, the G protein gets released. This thing is the first messenger. Now it can interact with something else, a second thing, and alter its structure or, have it released. And that's now the second messenger. And then that thing can go off and bind to something else and change its shape and alter its function. That's the third messenger. And then that one can go off and, interact with another thing, and that's, change it, and that's the fourth messenger. So you get these, what are called biochemical cascades that are initiated by the binding of neurotransmitter in the synaptic cleft to the metabotropic receptor, and that release of the first messenger, or the G protein. And, again, there are different G proteins. So this one is connected, it's going to respond to this chemical and set off this biochemical cascade. This one's going to go to a different chemical and set off that biochemical cascade. And what's particularly powerful about these biochemical cascades is that they ultimately can activate, messengers, chemicals that will enter the nucleus, that can bind to your chromosomes, they can bind to the DNA, they can unwind or unzip, certain segments of the DNA and allow for transcription and translation of specific genes, or they can do the opposite. They can result in the winding up and the reduction in transcription and translation. So it's a really fascinating and extremely well conserved in an evolutionary sense. Like these biochemical cascades go way back, in terms of evolution, to fruit flies and C. elegans, etc. But it's a way for neurons and other cells too, actually, to listen to the chemical, activity in their surrounding environment and respond, link that back to changes in gene expression which grow and alter their very structure, so that they can respond differently and hopefully, potentially more effectively in the future. So big changes in synaptic activity, right? Like more and more neurotransmitter, more and more stimulation will result in a lot more, G proteins being released, a lot more second messengers activated, third messengers, etc. A lot more changes in gene expression. The same is true if there's a sudden reduction in activity, at a synapse, and there are fewer G proteins sort of being released. There can be, also compensatory changes that occur in terms of not only the electrical charge difference across the membrane, but changes in gene expression that result from that, drop in activity. So metabotropic receptors are, really quite fascinating and important, as a class of receptor in the nervous system. Another terminological issue is that many labs, many scientists don't use the term metabotropic receptor. That's like the effort to sort of standardize, but they use a different term. They call these G protein coupled receptors because they are, before the binding of a neurotransmitter, coupled to a G protein, right? When the binding of the neurotransmitter results in the release of that G protein. So you'll hear the term very commonly in research papers and in laboratories and at conferences. When people talk about this particular class of receptor, they call them GPCRs, G protein coupled receptors. But that's another name, another term for metabotropic receptors.